Oops. Cool. All right. Well, let's get started then. Hopefully this mic work. Yep. Okay. All right. So today we're going to continue our neuropharmacology series, lecture series. So yesterday we had a bit of a introduction, kind of expanding on the kind of concepts that we talked about in PHAR3320 in the first semester. So today and in the next few lectures, we're actually going to focus on some actual neurological diseases and disorders and kind of how we can use neuropharmacology as a therapeutic tool and kind of how do these therapies actually work. And today we're going to focus on depression and how antidepressants work in a broad sense. So what do I want you to get from this lecture? Well, I know that this isn't a neuroscience unit, but I still think it's important to understand a tiny bit about the pathophysiology of depression. So when we talk about pathophysiology, we're talking about what are the changes in the body or the brain that accompany depression. We're not going to go into it very, very deep, just very superficially, but I think it's just important to understand. And then we'll move on to antidepressant treatments. So we'll talk about common pharmacological treatments of depression and their mechanism of action. Mostly we're going to focus on these three antidepressants, so monoamine oxidase inhibitors, tricyclic antidepressants, and SSRIs, which is probably the most common antidepressants prescribed. And then we're going to briefly touch on alternative antidepressant treatments as well, where we'll talk about ketamine and how that might be used in place of these monoamine-focused antidepressants. So I just want to preface this lecture by saying that, again, we're talking about depression in this lecture, which can, it can be a bit difficult to hear if you have a lived experience with depression. But I just want to say that everything in this lecture is just for educational purposes. It's not clinical advice. So if you do need to talk to someone or if some of this information you want to learn more, please, you can talk to UWA, has an after-hours support helpline. You can also talk to your GP. And I do also want to say a brief trigger warning. In the first two slides, there will be mention of suicide, just because it's part of the DSM criteria. So just a warning there. It'll just be in the first two slides, and then we won't talk about it again. Okay, so first of all, what is depression? I'm sure everyone here has heard of depression or they're aware of depression. It's actually one of the most prevalent mental health disorders that's experienced by individuals. It's estimated that approximately 5 to 10% of the population is living with depression. We think that that might also be underrepresented because depression shares a lot of kind of similar symptoms to a lot of other mental health disorders and also neurological diseases or disorders. And again, there is a bit of a stigma with depression as well, so people might not be seeking treatment or diagnosis. So we think that people living with depression might actually be higher than that 5 to 10%. But it's also estimated that approximately 20% of people, and again, the numbers differ depending on age, sex, and which country you're living in, but it's estimated that approximately 20% of people will experience at least one depressive episode in their life. So they experience the symptoms of depression, but it's only acutely. So it's a very significant disease. It affects millions of people worldwide, and there really is a lot of research looking at improving the lives of people living with it. And also interestingly, and it's something that a lot of people probably think about, is that since the start of the COVID-19 pandemic, there's been a very significant increase in the prevalence of depression as well. And there's a lot of kind of reviews, systematic reviews talking about this, where we're actually, since COVID has occurred, especially in the people that have experienced COVID and then had post-COVID symptoms, there's been a lot of increase in depressive symptoms in those people. And then just in society in general, there's been an increase in the prevalence of depression, especially in adolescence as well, where a lot of children were not able to go to school, socialize, they were stuck in the house, a lot of, hearing a lot of, fear-mongering, media reports, things like that. It's really affected people significantly. So it's something that does need to be kind of tackled head on. We'll talk about most of the symptoms, but commonly what we see in people living with depression is that they experience anhedonia, which is a reduced ability to experience pleasure, and also feelings of hopelessness and worthlessness. They're kind of the common mood symptoms that you see, but there's also a lot more that they experience as well. And the reason why this is such a big issue, apart from the interpersonal costs, is that this also represents significant societal and economic costs as well. So there's a lot of costs associated with things like loss of employment, so being unable to work, maybe having carers or family taking care of you, healthcare costs, things like that. This all represents a very significant interpersonal but also societal cost as well. And obviously this, some of these treatments, or some of these symptoms can lead to lifelong disability as well. And touched on it briefly, but the depression has very high comorbidity with other mood disorders and neurological diseases as well. So it's not just something that exists in its own vacuum. There is a lot of comorbidity with other factors as well. So what actually is depression? How is there a diagnosis of depression? Well, the DSM-5, which is kind of the manual of all kind of diagnosed mental health and other illnesses, outlines the following criteria to make a diagnosis. I won't go through it all here, but basically you need to be experiencing some of these symptoms. So things like a depressed mood most of the day, markedly diminished interest or pleasure in many activities, so that anhedonia, slowing down of thought and reduction of physical movement that is observable by others, fatigue, feelings of worthlessness, diminished ability to think, and then also increase in suicidal ideation as well. And a depression diagnosis is made when these symptoms cause the individual clinically significant distress or impairment, and again in those occupational or societal outcomes as well. So again, there's a lot of symptoms that's associated with it. Also other things that aren't talked about as well is that this might also lead to insomnia as well, and there's a very high comorbidity with anxiety disorders as well. So what is actually occurring in the brain that's causing depression? Well, unfortunately there's no real agreed upon underlying mechanism of why depression occurs. There's evidence from multiple systems that are implicated in the disease that we're going to touch on now. So we see evidence for changes in neurotransmitters in the brain. So we see that there's alterations in neurotransmitter activity. So specifically in the monoamines, we see that there's irregularities or alterations in the serotonergic system, dopaminergic system, and the noradrenergic system. So all these monoamines, some of these are really important for transmission, reward pathways, mood pathways as well. So we see that there's an alteration in these neurotransmitters that are responsible for those outcomes. Historically, we kind of think of this as the monoamine theory of depression, and we'll touch on it later. But basically, all these neurotransmitters are monoamines, and we usually see that there's some alteration in them. But also bear in mind that neurotransmitters can also be excitatory and inhibitory, and we do see some changes in the excitatory neurotransmitters as well, like glutamate. So we also see some changes broadly in neural activity as well, so just the communication in different areas in the brain. Again, some of those neurotransmitters that are implicated are the excitatory and inhibitory neurotransmitters, so glutamate and GABA, so kind of that inhibitory-excitatory balance in certain brain regions that are responsible for some of these moods might be affected. And we know this because if we alter neural activity with drugs or even brain stimulation, this can actually have an antidepressant effect as well. So it's not just increasing monoamines, but if you change the activity of certain neurons in certain areas of the brain, you can induce antidepressant-like symptom or antidepressant outcomes. So what else is implicated? We know that plasticity and brain connectivity is also implicated with depression as well. And we know this because some antidepressants actually induce changes in plasticity. So things like some SSRIs like fluoxetine, we actually see that this can increase the amount of dendritic spines. So remember those dendritic spines that I showed you yesterday, those little kind of finger-like knobbly projections that create those synapses? We see that when you take these antidepressant treatments and even with other things like exercise, you can increase the amount of those dendritic spines. So we're creating more synapses between neurons. They're making more connections. So this might be kind of what's responsible. Maybe these neurons need to make more connections. We see changes in BDNF. I don't know if you've heard of BDNF before. It's called brain-derived neurotrophic factor. It's a really big neuroprotective factor or protein in the brain. It's also really important for plasticity. We see changes in BDNF with antidepressants. They increase BDNF typically. We also see things like changes in neurogenesis. So we see with antidepressant treatments, they usually increase the amount of neurons being born in the brain. So this might be some of the therapeutic mechanisms because it taps into these. And also, if we use fMRI imaging, you've probably heard of MRI imaging before, magnetic resonance imaging, where you get put in this big giant machine that basically scans you using magnets and you can see into the brain. If we do functional MRI imaging, so you kind of look at the blood signal going to different parts of the brain, we use this as like a correlative activity. So if we see one area of the brain has more blood associated with it, and another area of the brain has an increase in the blood signal at the same time, we usually say that those areas are connected with each other. And we see that when people take antidepressants, you see changes in those areas of the brain that are active at the same time. So it seems to suggest that there's some plasticity. These areas are kind of communicating more with each other. They're creating more connections. And then there's also a really large amount of literature that I'm not going to get into talking about genetic and environmental factors as well. So there's evidence for genetic mutations and increases in depressive symptoms. You might have genetic mutations that increase the breakdown of monoamines, for example. Maybe you have genetic mutations that break down drugs or some of these antidepressants as well, which might play a part. Stress is a really big correlate of depression and anxiety disorder as well. So high levels of stress, especially during development, is a really large risk factor for depression as well. Also, diet is seen to play a really big role in depression as well. You've probably heard of the gut-brain axis before, where things that you eat, the microbiome in your gut can actually affect the way that your brain functions, and some of these might lead to depressant or antidepressant-like effects as well, but we won't go into any of that. So the treatment plan, I'm just going to briefly touch on this. Usually you receive it, if you are living with depression, you've received a diagnosis, and then usually put on pharmacological treatment, so usually that would be antidepressants. Usually you have to take these antidepressants for a few weeks because they take a while to induce a therapeutic effect. But a lot of times people can fail these antidepressant treatments, and there's so many antidepressant treatments out there that some of these diagnoses and treatments can actually take a really long time to kind of start producing a therapeutic effect, because if you spend 3 weeks on one antidepressant, then it doesn't work. You have to go to another one, spend another 3 weeks. It can be a really exhausting and fatiguing process. And a lot of the it's not just pharmacological treatments. A lot of times you'll be suggested to use psychological therapy as well, and that works in tandem with pharmacotherapy to kind of help you kind of retrain your mind or be able to live with the symptoms. And if all of these are unresponsive, usually your depression will be described as treatment resistant, and then you'll go on to other forms of treatment as well. So things like non-invasive brain stimulation, but then also electroconvulsive therapy, which is reserved for kind of really extreme cases of depression, which actually helps a lot of people, but it's kind of like a last resort treatment. Anyway, so moving on to the pharmacology. So I mentioned before that kind of one of the oldest theories for depression was this monoamine hypothesis of depression. So this predicts that the underlying neurophysiology of depression is a depletion in the levels of monoamines, like what I spoke about earlier, serotonin, norepinephrine, and dopamine. So there's neurons in the brain that produce and release these monoamines. So usually you'll have dopaminergic neurons or serotonergic neurons. They're present all throughout the brain. A lot of them are kind of clustered around areas of the brain that's important for mood and reward. So, you might see lots of dopaminergic neurons in the striatum or the ventral tegmental area, for instance, and a lot of serotonergic neurons are around those areas as well, and we usually see alterations in them. And so what happens is that these neurons, they produce these monoamines, This is just an example here of one presynaptic neuron, one postsynaptic neuron. This neuron might be releasing monoamines, so this one here is releasing, let's just say, dopamine, serotonin, and norepinephrine. The neurotransmitters, it receives an action potential, releases those neurotransmitters. Those bind to their postsynaptic targets and also their presynaptic targets, and they also get reuptaken back into the neuron for recycling. If the neurotransmitter binds to its own receptor, we call that autoreception, and this autoreception is really important for inhibiting further release. So just by, again, just thinking about the amount of neurotransmitters that get released into the synapse or the enzymes that eat it up, you can see that this would cause, alterations in a lot of things like mood if these neurons are responsible for it. So I just mentioned it, but monoamines are reuptaken by transporters as well. So the presynaptic neuron takes up the neurotransmitters that it releases, and then once they get reuptaken, they can be repackaged into vesicles again, so they can be released. So in this way, they get released, reuptaken, repackaged, released, reuptaken, repackaged. But some of these neurotransmitters as well also get broken down by monoamine oxidase. And monoamine oxidase is just an enzyme that breaks down monoamines into its constituent products. So we'll talk more about some of these mechanisms later, but just know that, again, neurons release these monoamines. They can affect their postsynaptic targets, or they can be recycled to be used again or broken down. So one of the big monoamines that is involved with depression and antidepressant treatments is serotonin, or 5-HT. And this is an excitatory amine neurotransmitter, and it's very, very important for mood and also eating habits as well. So serotonin is synthesized from 5-hydroxytryptophan, or 5-HTP, and this makes 5-hydroxytryptamine, or serotonin, 5-HT. And this is just showing you it here. So this is just showing you all the precursors. So tryptophan gets turned into 5-hydroxytryptophan by these enzymes, and then you have your serotonin here. And then, as you can see, monoamine oxidase breaks down serotonin into its aldehyde counterpart. But serotonin is implicated in many mental health disorders. It's not just depression. It's also implicated in anxiety as well. A lot of other anxiety disorders like post-traumatic stress disorder. I think OCD has links to serotonin. I think bipolar as well. There's a variety of different mental health disorders. And it's thought that the concentration of serotonin is directly correlated to levels of depression and anxiety, where usually it's said that a decrease in serotonin is linked with higher levels of depression and anxiety. So usually we try to increase the amount of serotonin to overcome those symptoms. And serotonin has many different receptors. We're not going to get into all of them, but just know that there's many different subtypes of these receptors. So for example, you got the 5-HT1A, so this is the 1A subtype. You got the 1B subtype. A lot of these are the autoreceptors, so they're present on the postsynaptic neuron, but also on the presynaptic neuron. So affecting these would inhibit the release of neurotransmitters. But then you've also got 5-HT2, 5-HT3. Most of these are G protein coupled receptors, except 5-HT3, which is an ion channel receptor. And again, they're expressed pre and postsynaptically, and they all have different antidepressant-like effects depending on if they stimulate the postsynaptic neuron or stimulate the presynaptic neuron. But don't worry too much about this. I'm just trying to show you that there's a lot of kind of targets that these drugs can affect. So a drug, for instance, can affect the 5-HT1A receptor, whereas another drug might affect all 7 subtypes. So it's just something to keep in mind. So then, if we know that monoamines are involved, we know that serotonin's involved, how can we actually start producing an antidepressant effect? Because usually we want to increase the amount of serotonin. Well, one target is that monoamine oxidase. So remember, monoamine oxidase breaks down monoamines after they've been reuptaken. So if we prevent the breakdown of those monoamines, maybe we can have more present in the presynapse, so more is released during stimulation. okay? So you're just stopping the breakdown of those serotonin neurotransmitters, so more is available, so you can have a larger effect. That's essentially what these monoamine inhibitors do. So they inhibit the enzymatic breakdown of monoamines to increase their bioavailability. There's more available to bind to their receptors. And what this does is that it increases the monoamine concentration, so it's more available at the presynaptic terminal. So then when an action potential occurs, you have more neurotransmitters that are available to bind to it. And there's two different types of monoamine oxidase. So you've got monoamine oxidase A, monoamine oxidase B. They're all differentially distributed throughout the body. They all break down monoamines, but as you can see in this cartoon, some monoamine oxidases are kind of more important for breaking down things like serotonin versus other catecholamines as well. So depending if your drug affects monoamine oxidase A or monoamine oxidase B or both, you're going to get different kind of antidepressant-like effects as well, and kind of their effects on their drug targets. So there's about 4 FDA-approved monoamine oxidase inhibitors. Some of these names include these ones here, so isocarboxazid, phenelzine, selegiline, and tranylcypromine. Yeah, I don't enjoy pronouncing those names. But one interesting kind of side effect of monoamine oxidase inhibitors, and I don't know if I talked about it in this lecture in first semester or I talked about it in another one, but you have this interesting effect called the cheese effect. I don't know if any of you have heard of this before, but basically if you're someone that is on monoamine oxidase inhibitors and you eat foods that are high in triamine, so triamine, as you can see here, is a monoamine as well, what happens is that if you're blocking monoamine oxidase, that means that you're going to get an accumulation of different monoamines, and one of these is triamine. Triamine's really present in foods like cheese, fish, fermented fruit as well. So these have high levels of triamine. So if you're blocking the breakdown of triamine, you get an accumulation of triamine, and what happens is that this can actually induce a hypertensive crisis in people as well. So you can actually have things like heart attacks if you're on monoamine oxidase inhibitors and you eat some of these foods. So there is kind of side effects with this, and you have to avoid certain foods if you're on these drugs. The other thing I wanted to mention was that most of these monoamine oxidase inhibitors are taken orally, so with a drug, but selegiline can actually also be applied transdermally as well, so you can have a patch that induces its effects as well. Okay, so that's one class of drugs that we can use to increase the amount of monoamines to have an antidepressant effect. We can inhibit monoamine oxidase, so prevent the breakdown. But what else can we kind of target that might produce an antidepressant effect? Well, one thing we can do is affect the membrane transporter. So again, remember that this is neurotransmitters are released into the synapse, and then they're reuptaken back into the neuron by the transporter. So if we affect this transporter in some way, we can change the amount of synaptically available monoamines. And that's what this other class of drugs does, called transport inhibitors. So monoamine reuptake inhibitors, they bind to this membrane transporter, and what it does is it prevents the reuptake of these neurotransmitters back into the presynaptic neuron. So if they're unable to be reuptaken, you have more of them present in the synapse, they have more time to bind to their postsynaptic receptors, and then you can get this antidepressant-like effect with its application. So it's doing kind of the same thing as the oxidase inhibitors, but through a different mechanism. So oxidase inhibitors stop the breakdown, so more is released during an action potential, whereas the transporters, after it's been released, prevents its reuptake, so it stays in the synapse for longer. So they effectively do similar things. You're increasing the amount of monoamines or serotonin, but they're doing it through slightly different mechanisms and through different receptors as well. And one of the most common form of these reuptake inhibitors that you'll hear is selective serotonin reuptake inhibitors, or SSRIs. And these are a class of monoamine transport inhibitors that prevents the reuptake, mostly focused around serotonin, although it is a little bit non-specific, but mostly it focuses on serotonin. So it works to increase the amount of serotonin at the synapse, which again, we have already talked about that changes in serotonin levels might be responsible for depressive symptoms. Now, interestingly, SSRIs, and I mentioned it at the start, they don't really have an immediate therapeutic effect. So once you start taking them within, a few hours or a few days, they generally don't have a therapeutic effect. So if you do experience an effect taking these drugs, it's most commonly a side effect of taking the SSRIs, so they're inducing some immediate changes, but you need to take these SSRIs for a long period of time. It's about 2 to 3 weeks before you're expected to see a therapeutic effect. Again, which can be, adds to the fatigue of the diagnosis and the treatment, and then also a lot of people looking for other kind of drugs that want an immediate effect as well. And this here is just taken from a paper. It's just showing you the serotonergic receptors in the brain and kind of where they're located. And this is done through PET imaging. So I don't know if you've heard of PET imaging before, but basically you introduce a radioactive tracer, a ligand, into the body. So you put a radioactive tracer in the body that recognizes this receptor, binds to the receptor, and then you do some imaging where you can basically look at the amount of the radioactive isotope in the body, so you can get a really quick snapshot of where these receptors are, and then your body just metabolizes the radioactive tracer very quickly. But this is what they did in this study, so it's just looking at different areas of the brain and where these serotonergic receptors are located. Where there's more red, it means that there's a higher density of these receptors compared to the blue. But as you can see, a lot of these receptors are just present all throughout the brain, mostly in the cortex, but also a lot of subcortical areas. So more are present towards like occipital lobe, some are more in the temporal lobe, some are more located, in the frontal lobe here, prefrontal cortex. So it just goes to show you that when you take these SSRIs, you could be affecting serotonin throughout the whole brain, not necessarily in those areas that are just affecting mood. It could affect many different brain regions, which adds to the complexity of the treating the disease as well. Now, the most common SSRI that you've probably all heard of is fluoxetine. This is an SSRI, and it's a highly active blocker of the serotonin transporter. Now, fluoxetine lacks affinity for many neurotransmitter receptors, but it can also act on other transporters as well. So its effect is not just at those serotonin transporters, it can actually affect other monoamine transporters as well. But some studies suggest that it's about 20 to 1,500 times more selective for inhibiting that serotonin over other monoamines like norepinephrine, but it still does affect those receptors. And I've listed it here, but there is evidence for minimal binding affinity for other postsynaptic monoamine receptors. So the adrenergic alpha-1 receptor, alpha-2 receptor, beta receptor, can also bind to histamine receptors, muscarinic receptors, also dopamine receptors. And this is a study that talks about it as well. So it talks about that it can also increase norepinephrine and dopamine levels as well. So even though it has low affinity for other monoamines, it might also be affecting these other monoamines as well. So it just goes to show you that, again, it is complex, and again, we might be affecting these other systems as well. SSRIs, especially fluoxetine, doesn't stimulate the presynaptic release, so it doesn't increase the amount of neurotransmitters released from the presynapse, it just stops their reuptake. So again, just demonstrating that mechanism. And mostly it affects those presynaptic transporters. There is evidence that it affects postsynaptic serotonin receptors as well, but we think that this is minimal. We think that most of the effect is due to blocking that reuptake transporter. So kind of related to SSRIs, there's some other kind of newly developed SSRI-like drugs. These are called serotonin modulator and stimulator drugs. And what they do is that they simultaneously modulate one or more actual receptors and also inhibit reuptake of sodium. So it's a bit more of like a specific action. So it's not just blocking that reuptake, it blocks the reuptake, but also blocks some of those postsynaptic receptors. Well, why would you want to do that? Well, these were developed because, again, I showed you that there was many different types of those serotonin receptors, and not all of these are involved in the antidepressant effect of the SSRIs. And many of these serotonin receptors can actually carry some unwanted side effects. So these were developed to kind of block those unwanted side effects while still increasing the antidepressant effect of serotonin. And this is just showing you, again, some of those different subclasses of the serotonergic receptors. Again, you can see some of these are G protein coupled, some of these are ion channels, some of them cause stimulation, some of them cause inhibition. And again, like I showed you in that previous PET diagram, they're all distributed through different areas of the brain as well. And one example of this drug is vortioxetine. So this is a serotonin reuptake inhibitor, but it's also an agonist of the 5-HT1A receptor, an antagonist of the 5-HT3 and 5-HT7 receptors. So a little bit more niche and kind of more selective in its abilities. So it reuptakes serotonin, so it keeps more serotonin in the synapse. It agonizes that presynaptic 5-HT1 receptor. Remember, this receptor is an autoreceptor, so it stops the release of serotonin. So it works on that mechanism. And it also antagonizes this 5-HT3 and 5-HT7 receptors, because it was found that by stimulating 5-HT3 receptors, it can actually induce nausea, vomiting, and GI problems, as well as that HT7 receptor. So basically what it's doing, it's increasing serotonin, but also blocking those receptors that might be responsible for some of the side effects. So again, just a newer class of drug that is a bit more specific in its action. All right, so moving on, another class of antidepressants that we'll briefly touch on are tricyclic antidepressants. You've probably heard of these. These are one of the more kind of older antidepressants. And these are a subclass of drugs that's based on this 3-benzene ring molecular structure that it has. And it was named after this because at the time this drug was found, its function wasn't really known. So they classified it based on its 3-benzene ring structure. So that's why it's tricyclic antidepressants. And these are similar to those SSRIs. They act on monoamine reuptake transporters, and they also act on receptor proteins as well. So what have these been demonstrated to do? Well, they've been known to inhibit presynaptic norepinephrine uptake transporters and also inhibit presynaptic serotonin reuptake transporters. So again, similar to an SSRI, but it has more of an effect on norepinephrine as well. So it's working on multiple monoamines. But it's doing essentially the same thing. So it's just increasing the synaptic concentration of monoamines. It's just doing it to different monoamines in different combinations as well. But there's several kind of side effects tied to tricyclic antidepressants. So some of these that we see is that it blocks postsynaptic adrenergic receptors, which might be responsible for dizziness, because it can it can induce low blood pressure. Because again, noradrenaline is really important for pumping of the heart and maintaining the circulatory system. It blocks postsynaptic muscarinic receptors. Muscarinic receptors, or acetylcholine, is really important for functioning of memory and the hippocampus. So if you're blocking these receptors, you might be affecting the acquisition of memory. But it also can block postsynaptic histamine receptors, so we can induce drowsiness as well, which is similar to taking an antihistamine. You might feel drowsy. So again, it's just an alternative antidepressant treatment. Usually the first kind of stop of a depression diagnosis is you'll usually be prescribed an SSRI first, something like fluoxetine, and then you will move on to kind of some of these other drugs depending upon your own individual needs and variability. So we just talked mostly about monoamines and how we can alter the levels of monoamines. But there's a lot of issues with kind of using and just focusing our antidepressant treatment on monoamines. Again, we still don't know what causes depression. We think it's linked to monoamines, but we know that with these antidepressant treatments, not everyone responds to this. And so this pharmacological intervention of increasing monoamines doesn't always lead to remission. And let's just say we have 4 people here that are living with depression. They go on this pharmacological treatment. There's estimates that approximately 30 to 50% of people actually don't respond to conventional pharmacological treatments, which is really sad and concerning. And this might be for a variety of different reasons. It could be the specificity of drugs, could be the way that they target the different parts of the brain. There's a lot of individual variability as well, different genetic information, enzymes that break these drugs down, different distribution of these proteins in the brain. So not everyone responds the same. And unfortunately, estimates up to 50% people don't respond to this. And it's really interesting because even though these interventions that we talked about, they increase monoamines and neurotransmitters, again, this doesn't always lead to improvements in depressive symptoms. And it's interesting because the timescale of these changes in monoamines doesn't really link up to what we expect the antidepressant effects to do. So let's say you take an SSRI, as soon as you take it, it should be blocking those reuptake inhibitors, so you should be getting more neurotransmitters released. But you don't see a therapeutic effect until 3 weeks later. So why are you not seeing a therapeutic effect even if you're increasing those serotonin neurotransmitters or norepinephrine? Why do you need to wait for 3 weeks? So again, there's evidence for this monoamine theory, but we think that there might be other kind of mechanisms at play. So what other alternative treatments are available that might be able to kind of overcome this limitation with these monoamines? And one of these might be actually looking at glutamate. So again, we talked about glutamate in first semester. Glutamate is the main excitatory neurotransmitter in the nervous system. So usually glutamate's present all throughout the brain. A lot of excitatory neurons release glutamate to cause excitation. That's how you get that transmission of the action potential usually. And we talked about it in first semester, but there's different categories of these glutamate receptors. So you have ionotropic ligand-gated ion channels, and you also have metabotropic G-protein coupled receptor channels. So glutamate can bind to both of these. Usually the ionotropic receptors causes a very quick, immediate excitatory response, whereas the G-protein coupled receptors take a little bit longer, and they usually induce transduction mechanisms to kind of change the probability of channels opening, secondary messenger systems to increase things like synaptic plasticity. Don't worry about it too much. But there's actually been a lot of kind of recent-ish, although it's a little bit old now, but a lot more research going into it, looking at levels of glutamate in the brain or in the body in people living with depression and people without, and seeing if there's changes. And there's actually a lot of supporting evidence for changes in glutamate. Now, depending on which brain structure you look, you see increases and decreases. But generally, what we see is that people with depression seem to have an increase in this excitatory glutamate neurotransmitter in certain parts of their brain. And we can do that by looking at, again, magnetic resonance spectrometry. So we look, we put, similar to MRI, we use magnets to image into the brain, and then this, by inducing this magnetic field, you get kind of these proteins in your brain wobble or resonate at a certain frequency. And if we know what that frequency is, we can image that protein. So for example, glutamate resonates at a very predictable frequency, so we can actually start looking at where is glutamate located in these different brain regions. And we usually see that there's an increase in glutamate in people living with depression. So there seems to be more excitatory neurotransmission. So again, people are looking at this as a potential biomarker for depression, but then also, can we target glutamate or the glutamate receptor to kind of bring those glutamate levels down to possibly induce an antidepressant effect? And again, this is just the NMDA receptor. I showed this in the first semester. But the NMDA receptor is a receptor for glutamate. It requires glutamate to bind to it, glycine to bind to it, as well as a voltage sensor. It needs to remove this magnesium pore. But if we have a drug that kind of blocks the action of NMDA, we might be able to bring those glutamate levels down or the excitation down. And one drug that's actually been demonstrated to do that, and is actually going through a lot of clinical trials at the moment, is ketamine. So ketamine is a short-acting dissociative anesthetic. It's used a lot in humans, but mostly in animals. You probably heard of it referred to as horse tranquilizer. It's gained popularity as a recreational drug in the mid-1990s, so a lot of people take a sub-anesthetic dose for recreational purposes. And what this drug is, it's a derivative of phencyclidine, which is PCP, and this is a non-competitive NMDA receptor agonist. So it actually binds to the NMDA receptor and antagonizes it. So it stops that glutamate excitation because it takes the space of its binding site. It's non-selective for different subunits of the NMDA receptor, so it just seems to block a lot of the 2A NMDA receptor subunits. Now it has relatively low affinity and low selectivity as well, so it has been demonstrated to bind to other kind of muscarinic receptors, dopamine receptors, norepinephrine. So it does seem to also affect those monoamine targets as well, but its predominant effect is mostly on glutamate receptors. And many studies have demonstrated now that sub-anesthetic doses of ketamine have been shown to improve depressive symptoms. So an anesthetic dose of ketamine is approximately 1 mg per kilogram. So sub-anesthetic doses are less than this. So most of the studies use about 0.1 to 0.5 mg per kilogram. And usually this is administered IV over 40 minutes. So they give you this sub-anesthetic dose over a long period of time. And this is actually a review, kind of recent review, that's just looking at different applications of, or clinical studies that have used ketamine for the use of depression. If you're interested, look at that paper, but generally it's showing, or what they found is that ketamine is much better than a placebo than in improving antidepressant-like symptoms. There needs to be a lot more kind of random control studies comparing ketamine to fluoxetine, things like that. But at the moment, it seems to improve depressive-like symptoms in people that are living with depression when compared to placebo. But there needs to be a lot more follow-up studies, but it seems to be really promising at the moment. And this is at multiple time points, so this study goes into it, but you see antidepressant effects within a few hours after this sub-anesthetic administration. Usually there's a few dissociative effects within the first 2 hours of its administration, but after this time period you usually see antidepressant-like effects. And sometimes this can last, in this paper shows, for up to 2 to 4 weeks as well. So it seems to be pretty promising. But oral delivery doesn't really work for ketamine that well because the bioavailability is really small because our body metabolizes it very quickly. So that's why a lot of these studies use IV administration of ketamine. But there still is a lot of unknowns. A lot of these studies just use a single dose of ketamine. So we actually don't have a lot of evidence for repeated administration. So is repeated administration safe? Does it also have antidepressant effects? Maybe it induces, addiction or maladaptive changes. We still don't know. But then this also brings into question so these are studies, or treatments that are just targeting the glutamatergic system, the excitatory system. Remember, there's the inhibitory system as well. So is there other ways of stopping glutamate transmission, maybe by increasing inhibition? Don't really know, but it's just something to think about as well. All right, so that's it for the pharmacology. I just got one more slide, just talking about non-pharmacological treatments, if you're interested in it. I work in, most of my research is looking at these non-pharmacological treatments of depression. So I'm really interested in it. If any of you are interested, feel free to email me. We have a lot of research opportunities in this area if you want to go down that path. But if a lot of people don't respond to pharmacological or psychological treatments, they're usually referred to other alternative treatments. And one of these is non-invasive brain stimulation. So exactly what that means, it's just stimulating the brain from outside the brain, so you don't need to go into the brain, place electrodes, anything like that. And there's different ways of doing this. One of the most common and TGA approved and FDA approved is RTMS, or repetitive transcranial magnetic stimulation. So what happens is that you usually have this coil, and this coil is just placed above the head. An electrical current goes into this coil, and when the electric current passes into the coil, this creates a magnetic field. And this magnetic field passes through your skin, through your skull, and goes into your brain. And what this does is that it changes the activity of neurons in the brain. And this does it very quickly. As soon as the magnetic field goes through, it changes the activity of the neurons. So we can make them more excitable and start firing action potentials, or we can inhibit them and stop firing action potentials. And we can target this to different parts of the cortex. So we can apply it to the prefrontal cortex, which we think is important for depression. And we can, induce plasticity in this part of the brain and cause an antidepressant effect. This stimulation is not impeded by the skull, so the magnetic field penetrates the skull really easily. The only thing that changes the strength is kind of the distance from your skull. So the further away you put it, the less magnetic field's reaching. A kind of limitation is that the stimulation is mostly cortical, so the brain's pretty big, but most of the stimulation affects kind of the cortex, 1 to 2 centimeters into the brain. It doesn't really reach those deep brain structures that are important for mood regulation. But interestingly, even though it's stimulating the cortex, we see a lot of changes in those subcortical areas with TMS. So even though it's not directly stimulated by it, we still think it's getting changed by TMS. But TMS also requires a lot of specialized training. You need a really big kind of power supply and machine to do this, and you need someone trained to deliver it to the right area. So another form of stimulation as well that is not as promising as RTMS, but does have therapeutic effect, is TDCS, or transcranial direct current stimulation. It's different to TMS. In this way, you just put 2 electrode pads on the head, and then you basically just put a current through the electrodes, and that creates a voltage difference which excites or inhibits neurons. So it works similarly, but you're actually applying a direct electrical current rather than a magnetic field. Again, stimulation is not really impeded by the skull. It's also really easy to use. It's a nice kind of small portable device which might lead to potential home use. It's not as efficacious as TMS, but it's still really promising, and again, another alternative treatment. And we do some work in TDCS as well, if you're interested. But one last one I want to touch on, which is kind of a new form of brain stimulation. It's getting a lot of traction at the moment, is ultrasound stimulation. So you're probably aware of ultrasound. We use it as a way to kind of like image the body. Basically, it just shoots really high frequency sound waves into your body, and then the receiver picks it up and you can image inside the body. But a lot of people are using this to stimulate the brain because instead of using magnetic fields or electricity, basically you put very high frequency sound waves into the head, and these sound waves can kind of, we don't really know how it works, but they kind of mechanically move the neurons, and we think that opens and closes ion channels, which changes their activity. But there's a few limitations to this. Well, one advantage is that it can reach really deep brain structures that RTMS or TDCS doesn't really do. So you can get a really specific stimulation of like a nice, very deep brain structure. But because you're putting sound waves into the brain, the skull is actually very porous, and so it absorbs a lot of those sound waves. So a lot of the signal gets attenuated by the skull. So you actually need to really calibrate your dose of ultrasound. And there's also a bit of a safety limitation. You actually get a lot of heat generation with ultrasound stimulation because, again, you're sending really high frequency sound waves into the body, which mechanically moves the proteins and the different neurons. And people use this in studies to actually lesion the brain as well, so it can create a lot of heat. But the therapeutic doses don't do this. I'm just saying that there is a limitation of a trade-off between frequency, deep brain structure, and heat as well. But again, there's a lot of work going into these areas because, again, there's 50% of people don't respond to pharmacological treatments, so these are alternatives. And there's a lot of space in here for research because there's so many parameters we can use to stimulate, and we still don't know the basic mechanisms. Anyway, that's it for today. So thank you for coming today. Again, don't worry too much about the neuroscience, just focus more on the drugs, so the monoamine theory of depression, the drugs, SSRIs, things like that. And if you're interested in depression or brain stimulation for therapeutic purposes, feel free to email me, because we do have a lot of research projects and kind of expertise in the field as well if you're interested. But thank you very much.